So it is, oh, okay. It's my pleasure to introduce Carla Saleh from Institut Pasteur. As you maybe noticed, the QBI Seminar Series has really been focusing on virologists as of late, and we got the tradition continuing here with Carla. So it's great that she's here. She's done such great work in a number of different areas in the virology world, looking at, for example, RNAi immunity in species like Mosquito and Drosophila, and just really done, really fantastic mechanistic work in antiviral biology in these species, and also looking at defective viral genomes as potential therapeutic tools, collaboratively with Marco Vignuzzi, who gave a talk here recently, and just some of her background. So she's got her bachelor's and master's from Argentina, and then she got her PhD from the Pasteur Institute, and then came to UCSF and did her postdoc with Dr. Andino here, and then went back to the Pasteur Institute, where she's been a professor for a few years, 15 years. Wow.
I know, only 2.
Full professor, 2 years. She was actually telling me when she was here, I guess you were here when you moved over from Parnassus, right? In 2003. She was telling stories about how it was kind of dodgy around here, where you needed groups of people to go to your car. But I think you were safe at the end, right? So it's very different now, as you can see. And she's got numerous awards. I won't mention them all. I'll just mention one. She became an EMBO fellow a couple of years ago. So she's done a really great job in Paris over the last 15 years. And she's here on this trip. We're exploring a new relationship with Institut Pasteur and QBI. So the goal is that should be a bridge between these two places as we try to get a presence here in San Francisco with respect to the Institut Pasteur. So we're exploring that, so more to come on that. And we're delighted to have you here to hear about your very exciting work.
Thank you, Nevan, and thank you everybody that is online or that is here in person. I must say that I am very moved. I think I never was in this room as a PI. I always was here as a postdoc being evaluated. And I decided to be evaluated today and to talk about a total new area of research that we are doing in the lab and to get your feedback, guys. So thank you, Nevan, for the invitation. And today I want to talk, what do we do with mosquitoes that carry viruses and how we deal with these mosquitoes. So as you know, mosquitoes have very bad press. And normally if I ask who like mosquitoes, none of you will raise a hand, right? And it's with a reason, because mosquitoes are responsible for approximately 17% of all infectious diseases, and they cause more than 700,000 deaths annually. That at the shadow of SARS-CoV-2 is nothing with 18 million of deaths we have seen with COVID. However, when we think at global warming and how the mosquito range is expanding, we know that we're poised to see several outbreaks that will come from mosquitoes in the years to come. So the threat of emerging viruses and to predict future human disease that will emerge and spread is really very difficult. It's an endless challenge, and we still don't know how to do it. That is why we are in the current situation. What are the reasons that we are not yet able to predict outbreaks? One of them is that the majority of new infections are of animal origin. So here again, I will be concentrating this talk on mosquitoes, but you can extrapolate everything I am saying and think at the bats in China. So here we have the mosquitoes. Normally, the mosquitoes that are carrying viruses that we still don't know are forest mosquitoes or sylvatic mosquitoes, and they circulate in a cycle between mosquitoes and animals in the forest. And then sometimes there is a close contact between humans and these mosquitoes, and then the human get infected and come back to the city, where now we have urban mosquitoes that are the mosquitoes you know. You are normally never bite by forest mosquitoes. You are bite by urban mosquitoes, like Aedes aegypti, Aedes albopictus, that will maintain the cycle. So most of the animal infections, so most of the new infections are of animal origin. And when we think at viruses infecting wildlife, we know less than 1% of all these viruses. So the potential to have emergence of a new virus is enormous. And to that, we have the third problem, that is climate change. So global climate change is increasing the contact with animals and vectors. And regions that were before preserved for human contact, now they are in contact. So here you see, you have an example of albopictus distribution. So albopictus is the vector of, for example, chikungunya. That is in the year 60s. The map is not empty. There are some red dots here in the South Pacific and in the Indian Ocean that you cannot see. That is the current distribution. And that is the distribution in 2050, when the temperature will be 1.5 to 2 degrees higher than the temperature today. So you see that there is basically no region of the world that will escape to the presence of Aedes albopictus, that is an urban mosquito that likes to live among us, that is all the day active. So that is an example for Europe. You know that we live in Europe, that is France, I live in France. So I am very worried about mosquitoes. Aedes albopictus has already colonized most of Europe. So when we talk about mosquitoes, the first idea is to kill them. That is what we do at home, right? We kill mosquitoes. So why we just don't develop strategies that will eliminate the mosquitoes? It is possible to imagine a world without mosquitoes. And the answer is very simple, and the answer is no. Mosquitoes have an important role in the ecosystem at several levels. The evident one is, of course, the food chain, but there are more to that. There are at least 3,500 species of mosquito, but only 10% bite humans. So you cannot put, for example, insecticide that will not discriminate between the mosquitoes and kill all the mosquitoes, because only 10% bite humans. Mosquitoes are essential as pollinators. A lot of the food we eat is pollinated by mosquitoes. Mosquitoes are important food for birds. The larvae of mosquito that is in the water is food for hundreds of species of fish. The larvae also process detritus and help with the nitrogen cycle. So in the words of Anthony James at UC Irvine, it's better to deal with the mosquitoes we have now and try to fix them somehow so they will not transmit virus, than to eliminate them. Because we know also that nature has horror of emptiness. And if we remove the mosquito reservoir, we don't know what it will come next, okay? And we will not be ready to make face to what it come next. So the main problem with viral vector-borne diseases is the absence of vaccine, the emergence of insecticide resistance. So we are confronted to the need, like really an urgent need, to develop new methods to fight arboviruses in mosquito. So how can we tackle this problem? And that is what we have been trying to elucidate in the lab. So for the last 15 years, my lab has really dissect the immune response of the mosquito, the antiviral response of insects, Drosophila and mosquito, trying to understand exactly how the antiviral responses work in the hope that we can manipulate it to create mosquitoes that will somehow be refractory to infection. So one of these approaches, it has been to consider the multiparted interactions within a mosquito. So like all of us, the mosquito is also colonized by a microbiota. The microbiota is the bacteria, the virus, and the fungi that live inside the mosquito, and that they are good, okay? Let's use the term good versus bad. But it's the natural microbiome. Like we have a microbiota, mosquitoes also have microbiota. It has been shown that microbiota is essential throughout the life cycle of the mosquito. So microbiota is essential for larvae development. It's also essential for successful fitness in the adult, for reproduction. So microbiota really control the life cycle of the mosquito, and it will, the relationship of the microbiota with the larvae and with the adult, it will also be very important to determine the capacity of the mosquito to transmit later a virus. So manipulating the microbiota seems like a good strategy to try to get mosquitoes that will not get infected. And one of the best examples that you are all aware of has been the use of Wolvakia by the World Mosquito Program. So Wolvakia is an endosymbiont, and what the World Mosquito Program did was to colonize mosquitoes with Wolvakia and release them in the nature. And these mosquitoes have a reduced arbovirus transmission that has been proved for dengue, for Zika, for chikungunya, yellow fever, to the point that WHO has certified the utility of the Wolvakia strategy to control dengue in regions where dengue is highly endemic. However, we don't know exactly how Wolvakia produce this antiviral effect. There are a lot of studies, there are suggestions, there are theories, there are studies that show that lipids are involved, that oxygen stress is involved, but there is not a clear mechanism. And because we don't understand exactly what Wolvakia is doing, we cannot predict if mosquitoes will become resistant to Wolvakia. We cannot predict if viruses will learn how to escape Wolvakia either. So we are using a strategy that we don't know how much it will last or how long it will last. So again, it's imperative to develop methods that will complement the Wolvakia methods or new methods that will come over when Wolvakia is not useful anymore. So in the lab, we propose to manipulate the microbiota of the mosquito, in this case, specifically the virome and the bacterium, to develop ecology-friendly strategies for vector control. Basically, what we want to do is like if eating probiotics, right? We want to manipulate the microbiota and then give it back to the mosquito. So like we are treating the mosquito with probiotics that will be safe for everybody involved, and try to see, then we manipulate, and then we see if this manipulation is capable of stopping the virus, if we can achieve stable infection, heritability, because when you do that, you don't want to be doing manipulations every rainy season, right? You want to make a modification that then you release in nature and take over the population. So then we could do mass rearing and some kind of semi-field trial. That is the idea, okay? That is the hope. Today, I will show you our first steps and our first results into the manipulation of the virome and the bacterium of mosquitoes. So let me start with the virome. That is the work of a great postdoc in my lab, Cassandra Ko. Cassandra comes from Indonesia. She's in the third year postdoc in the lab, getting ready to become independent. Remember her name because I think she will do waves. So the virome of a mosquito is also what we call insect-specific viruses. Insect-specific viruses are viruses that are in the mosquito, but that cannot replicate in humans, okay? They are insect viruses. And metagenomic studies are revealing novel viruses every year. These insect-specific viruses, they belong to all the classes of viruses that you know. They are very diverse. And what is very interesting about them is that it has been shown that this insect-specific virus can influence vector competence of the host mosquito. So again, vector competence is the capacity of the mosquito to acquire, replicate, and transmit a virus, okay? And it has been shown that in presence of certain specific mosquito-specific virus, for example, numirin virus, you can block the infection of Zika virus. So the presence of a given insect-specific virus block the arbovirus. The arbovirus, now it cannot replicate in the mosquito, replicate to much lower levels, and then it cannot be transmitted. So here you have certain examples, different insect-specific viruses, and the arboviruses they block. Most of the time, the blocking is specific. Flavivirus block flavivirus. But we also have examples where flavis can block alphas or where flavis can block orthobunia viruses. So different families of viruses can block between themselves. How they block, it also remains a mystery. Several hypotheses. It could be because of homology of sequence. So some people postulate that RNA interference could be responsible for this blocking. It could be a super competition problem. It could be a competition for resources inside the cell. It's not clear the basis of the blocking, but what is clear is that the blocking exists. So because we have these good viruses that can block the bad viruses, we wonder if they can be manipulated to block arbovirus transmission, but in a rational way, right? So we went into virus hunting. What we wanted to do was untargeted DIM sequence that will help us to identify the circulating insect-specific viruses around the world, not just the colonies you have in the lab that have been for years, that have a microbiome that is totally reduced and characterized. We really wanted to go where our forest mosquito are, the sylvatic mosquitoes, and know the virus that these mosquitoes are carrying, and see if then we can find some of these viruses, correlate with epidemics, and find some of these insect-specific viruses that will have inhibitory capacities for arboviruses. To do that is very easy in paper, but it's really an odyssey. You have to hunt the mosquito, discover novel virus, identify seasonal and geographic trends, because all of that will be important for where you will apply the technology. Develop protocols for molecular identification of mosquito species, because when you go and get sylvatic or forest mosquitoes, not all mosquitoes have been identified phylogenetically. Develop transinfection protocols, and determine the outcome of this arbovirus, insect-specific virus co-infection. So it's really a titan work. So for the virus hunting, we work with the Pasteur International Network. You know, Pasteur is not only Paris. Pasteur, we are 33 institutes around the world. For this project specifically, we work with Institut Pasteur in French Guiana, in Bangui, in Cambodia, and in Madagascar. So all these regions were selected because they have a history of outbreaks for arboviruses like Zika, dengue, chikungunya, and also because they have very particular ecosystems. For example, Madagascar is an insular ecosystem. Bangui is in the middle of the forest, from where everything where everything has a lot of origin for disease over there. And then Cambodia and Guyana represent where we have the old world virus and the new world viruses, and also how the mosquitoes diverged when they went out of Africa. So we sampled mosquitoes. We did longitudinal collection and sampling. And here, just let me say that all of this work was done in pandemia. So I know that the people that did this work is not here this talk, but I just have to clap for them, because to keep doing this longitudinal sample during COVID was a nightmare. So we have the representation of dry and rainy seasons in each one of these 4 countries in several places that represent different biomes. So we have mosquitoes that come for forests, mangroves, coastal towns, agriculture, farming, urban or savanna. So everything is represented there. So here you have an idea of our sampling, so the dry and the rainy season in 2019 and in 2020, and the distribution of the most common species. So you can see always in rainy season, we have more mosquitoes, but mosquitoes do not disappear, of course, in dry season. You keep collecting mosquitoes in dry season. And the distribution is more or less the same. There is always a big abundance of culex here in orange, Anopheles in yellow, and Aedes in red. So once we collect all these mosquitoes, we develop protocols. And here, again, you need to think that to develop protocols, you need to sequence. To sequence, you need to know the RNA that you will deplete. But for these mosquitoes, the ribosomal RNA sequence is unknown and is not conserved. The ribosomal RNA sequence of an Anopheles is very different of the ribosomal RNA sequence of an Aedes mosquito. What is completely similar is the structure of the ribosomal RNA, but not the sequence. So there has been a lot of optimization and work going on there. And I can say that the day of today, this slide is all, at this day we have 95 viruses that we discovered. Today we have 160 viruses. So the ones that are in dark color here, they have a high similarity with things that were already described, but everything that is orange and yellow, these are new viruses. That is why in the names you see like. They are similar over some nucleotides or for the RDRP motif, but it doesn't mean that these are these viruses. So all of that are new viruses that we discover that are part of the virome of mosquitoes from different countries and of different species of mosquito. Here I show you an example of distribution. I think is always interest. These are urban mosquitoes, the mosquitoes that always bite us, Aedes aegypti, Aedes albopictus. So Aedes aegypti carry in dry or rainy season some known viruses like fuzzy charon virus and some unknown viruses. But there are several viruses that we could detect in Aedes aegypti. When you look at Aedes albopictus, your fair conclusion is, oh, Aedes albopictus, it doesn't have a virome. No, it's not true. Aedes albopictus has a virome, but it's so different that it's not similar to nothing that we know. So here we are really in virus discovery, because when you name a virus, you name it for what is similar to. And if it's not similar to nothing, you're missing it, right? So there is a lot of work that you have to do here, because Aedes albopictus, it also has a rich virome that is not similar to nothing that we already know. What happened with sylvatic mosquitoes? We have some very interesting trends that go with, for example, the seasons. Here you see Culex mosquitoes, and you see 3 different sites in Cambodia during dry season and during rainy season. And you see that, for example, the mosquitoes that come from Prague-Toll, they have Culex mosquito virus 6 only in Prague-Toll. So there is a geographical trend and a seasonal trend to insect-specific virus distribution in mosquitoes. And that is very important, because you could imagine that, for example, we have Zika in French Guiana, okay? And we have the same mosquito, but however, we don't have Zika in Southeast Asia, is an example. So you could imagine that the difference is because of the virome that the mosquito is carrying. And if we swap the viromes, if now I get the virome of the Southeast Asian mosquitoes and colonize my French Guiana mosquitoes with this new virome, perhaps they will become refractory to Zika, right? So that is the idea behind. So we wanted to have a proof of concept for that. And I will talk a little of transinfection, and I will show you an example. It's the example of Palm Creek virus. So Palm Creek virus is an insect-specific virus carried for a mosquito that is called Coquelettidia santogaster. So as I tell you, there are a lot of mosquitoes. So Coquelettidia santogaster is in Australia and carry this Palm Creek virus. So we isolate this virus, and we wanted to know if a virus from Coquelettidia was capable of infecting Aedes albopictus and Aedes aegypti. Why we were interested in doing that? Because some papers show in the past that Palm Creek virus is capable of inhibiting West Nile virus in Culex mosquito. So we wanted to know if we could put Palm Creek in Aedes albopictus or Aedes aegypti. This, taking a virus from one species and moving it into another species, is what we call transinfection, okay? So up to today, we don't know ways to infect with insect-specific viruses. In nature, insect-specific viruses are mostly transmitted vertically. So in the lab, we have tried several things. We have tried to inject, we have tried to feed with sugar, we have tried to feed with blood, we have tried to make osmotic stress or things, but for the moment, the only way we have to infect with the insect-specific virus is to inject. We inoculate the mosquito with the insect-specific virus. So what we did was to inoculate Aedes aegypti mosquitoes and Aedes albopictus with Palm Creek virus, and then we did the time course, and we checked for the presence of the virus in the carcass, in the midgut, and in the ovary. And why in the ovary? Because we want to be sure that the insect-specific virus we're putting is going to the ovary, because we have to have a chance to vertical transmission. If we want to develop a vector strategy that will be easy to implement, you need vertical transmission. So we saw that Palm Creek virus efficiently infect Aedes aegypti and Aedes albopictus in all the tissues and all the time points. So then we wanted to check if the presence of Palm Creek virus in Aedes aegypti and Aedes albopictus, it will be inhibitory for, I don't know what happened. Perhaps I keep seeing, no? No. We wanted to check if the presence of Palm Creek virus in Aedes aegypti or Aedes albopictus, it will be inhibitory for Zika and chikungunya, that are 2 viruses commonly carried by Aedes aegypti and Aedes albopictus. So what we did, we took mosquitoes, we inject Palm Creek virus, we wait 4 days to be at the peak of replication of Palm Creek, We did a viremic blood meal with our arbovirus, and then at 7 or 14 days post-blood meal, we collect saliva and dissect head, midgut, and carcass, titrate, and look at the effect of the presence of Palm Creek in the replication of Zika or chikungunya. So let's go fast over here. In green, you have Aedes aegypti. In blue, you have Aedes albopictus. Here you have Zika, chikungunya, and chikungunya. And basically, when you measure the virus in midgut, carcass, head, or saliva, you don't see any difference between the mosquitoes that carry Palm Creek virus or the mosquitoes that are mock infected. So Palm Creek virus has not effect on Zika or chikungunya virus transmission. It seems like a negative result, but it's not, because in reality, it's showing us that the interactions between insect-specific virus and arbovirus need to be examined on case-by-case basis, right? And as I mentioned before, we have exactly 160 viruses to check. So I hope the postdoc of Cassandra will not last 160 years, but I am sure that occasionally we will find the virus or the combination of insect-specific virus that will render a mosquito refractory to arboviruses. Because of the depth of our deep sequencing, it's very important to understand that when we sequence the mosquitoes that we have hunted in the world, we are able to see the mosquito that we sequence. In this case, for example, the example is Culex. We can also see the component of virus and bacteria that this mosquito have. And we can also see where the mosquito eat, right? In this case, we know that the mosquito bite a chicken because the mosquito that we sequence was a female that has blood in the belly because it has bite somebody. So here we see that this mosquito, this Culex mosquito, bite a chicken, probably bite a horse at some point here. And when we can look, we can also identify viruses and we can identify bacteria. And that opened the door to the second strategy we are developing in the lab, that is the bacteriome. So the bacteriome is, we started really recently. What I am showing is very preliminary data, is the work of a postdoc, Ottavia Romoli. Ottavia was first in the Institut Pasteur of Lyon, where she developed techniques to produce notobiotic mosquitoes. And now she's joined the lab to develop new strategies using the bacteriome to immunize mosquitoes. So again, the idea here is to immunize mosquitoes against arboviruses, and I need to take you a little over the RNAi. You know that the main antiviral response in insects is the RNAi response. And how is that work? Is that when the viral genome is replicated, it will generate double-strand RNA intermediates that are recognized as a danger signal and immediately taken care by dicer that will process it into siRNAs. And then these siRNAs will be loaded into Argonaut that will be used to degrade the target complementary RNA. So that is the way that the RNAi machinery will inhibit virus replication because it's degrading the viral gene. So using that, what we want to do is to take the bacteria, sorry, it's going too fast, there. To select candidate bacteria from the microbiome of our mosquito. It could be bacteria like Pseudomonas, Elisabethingia, Enterobacter, Chromobacterium, that are common bacteria inside the mosquito. We want to be able to manipulate these bacteria in order to force the bacteria to express double-strand RNA. Double-strand RNA against the arboviruses we want to fight. For example, dengue, chikungunya, Zika, Mayaro. Then with this bacteria, we can introduce it to mosquitoes because we have all the techniques to do notobiotic mosquitoes. And then this mosquito that is monocolonized, we can assess the effect of the double-strand RNA produced by the bacteria for the antiviral power. So as a proof of concept, we are not using the native bacteria in the mosquitoes because they need special ways for being cultured or for being manipulated. So as a proof of concept, we are using E. coli. The strain HT115D3, that is a strain that has a mutation, so it doesn't have exonuclease activity. So it has a plasmid that has 2 T7 promoters and terminators, and in the middle we insert a viral sequence of more or less 600 base pairs that will produce the double-strand RNA. And then we put this plasmid into the bacteria, right, that has the T7 polymerase under the control of a lag promoter. So we feed the bacteria culture with IPTG, and we should have expression of the double-strand RNA. So that is how we obtain our monocolonized mosquitoes. We get mosquito eggs, we sterilize them, so we obtain germ-free larvae. We add our bacteria culture, and we obtain notobiotic larvae, so larvae that is only colonized by the bacteria we introduce. We wait the larvae, the pupae, and the adult, and we check if the adult is carrying the bacteria, and we check also if the adult is having double-strand RNA inside. So these first results were very encouraging. Here you see the colonization success of adult mosquitoes that have eaten E. coli. So that means that the females were fed with sugar feed or blood feed. It doesn't make a difference, you see here. And they were fed only at the larval stage or at the larval and the adult stage. So in order to have adult mosquitoes colonized by bacteria, we have to feed them in the larvae and in the adult stage. And you see here, we have a beautiful colonization. That is bacteria supplemented in 10% sugar. And then we check also in our mosquitoes if they were producing double-strand RNA. And again, you can see here, that's a male, these are the females, sugar fed or blood fed. And again, they were eating bacteria supplemented with sugar, and they are producing double-strand RNA. So we don't know yet how this double-strand RNA will be able to get out of the bacteria. It will lyse the bacteria. There will be some kind of transport. We don't know. The proof of concept now is what happens if now we put the arbovirus for which we designed the double-strand RNA. So this experiment that is happening as we talk is like that. We have adults that are fed with the bacteria. Then we let them starve for 1 day. We let them starve, so then they are hungry and will take a lot of blood, right? So then we give them a Zika infectious blood meal. We keep adding bacteria because we don't want the bacteria to be loose, because the bacteria is bringing the antiviral molecule, the double-strand RNA. Then we dissect midgut carcass and head, and we do plaque assay and see what happened with Zika virus in this mosquito that was carrying refractory bacteria. So that when you invite me next year, I will tell you the results, because at the moment we don't know it, but we feel very hopeful with these experiments. So one of the key steps to produce new vector control strategies is that the modification that you will do will be inheritable, right? Again, because you don't want to be in permanence feeding mosquito or injecting mosquito and releasing new batches of mosquito. You want to make one modification that keeps going into the nature. So for the double-strand RNA, we expect to be inheritable. Why? So again, let me recapitulate very fast the antiviral response in mosquitoes or in Drosophila. We have viral RNA that will replicate and produce double-strand RNA that will trigger the innate antiviral response, generate siRNAs that will have a sequence-specific degradation. That is the RNA pathway that we all know. But the RNA pathway is more complex than that in insects, and in reality, what we have is that this viral RNA is reverse transcribed by transposons inside the cells and produce viral DNA. This viral DNA, these are all RNA viruses, right? So now we have a DNA form of an RNA virus. This viral DNA will undergo transcription and will create more double-strand RNA. So it's like boosting the innate antiviral response to have a better response that allows the mosquito to be persistently infected with the virus. So what we discover is that if we inject double-strand RNA against Symbis virus, that is also an arbovirus, in a female, in a virgin female, so the symbol here, the little horns at the top of the female, mean virgin. So a virgin female is injected with double-strand Symbis or double-strand control. Then we collect the embryos and bleach the embryos, And then we challenged the F1 with Symbis. What we discovered is that the F1 that the mothers of Symbis, the F1 is now protected against Symbis. So a little piece of double-strand RNA that was present in the fly, these experiments are in Drosophila, it was enough to confer protection to the F1. You see it here measuring luciferase. You see it here by PFUs compared to double-strand RNA control. But it was not only the F1, because, okay, a lot of things can go to the F1. Look at the effect. It was really maintained over the F2, the F3, the F4, the F5. So the great-great-great-grandchildren flies of the mother fly that saw a little piece of double-strand RNA inherit something that made this fly resistant to Symbis virus infection. So we check what they were inheriting. And here you have, is another example, is another virus of Drosophila. This is DCV from Drosophila C virus. So that's the mother. And you see the mother has active DCV replication. You see here by RT-PCR. And you see by PCR this DNA that I tell you that the virus produced, viral DNA. So then we took the embryos, we bleached, we obtained the F1, and we went to the F2 to be absolutely sure of what we were having in our hands. There is no more DCV replication, as you see here by the absence of a band, but look up, there is still DNA from DCV. So somehow the DNA of the virus that was infecting the mother is passing through generations. And that is also happening in mosquito. We have a mosquito that was infected with chikungunya. Then after 7 days, it was offered a non-infectious blood meal in order to get the eggs. The eggs were collected, hatched, we obtained adults, and then this F1 was challenged with chikungunya. And again, we saw inheritance of immunity. So mosquitoes, the F1 of mosquitoes, where the mother has been infected with chikungunya, they get less infected with chikungunya. So we know that inheritance is possible. We know that immune priming and that inheritance of an antiviral response against a given specific virus is possible. And that is why we think that our modification of double-strand RNA has a real opportunity to become inheritable in our mosquitoes. So to finish, what I have shown you today is preliminary results of methods that we are developing that we believe to be complementary to other intervention measures, for example, Wolvakia. We believe that these strategies can be rapidly adapted to variants and to emerging viruses, because imagine if it's so simple as changing the double-strand RNA. Today we have an outbreak of Zika, but tomorrow we have an outbreak of Mayaro. However, we have the platform ready and we only have to change the double-strand RNA. And these are sustainable strategies too, because as I show you here, that is an individual mosquito. And I know that in this mosquito, these bacteria live with these insect-specific viruses. So you can even imagine control strategies where you combine a given insect-specific virus with a given bacteria in order to obtain a mosquito that is refractory. And again, it's only using probiotics. So are we going to be able to do semi-field trials? Probably, because there are already big wins. What I show you is that we were capable of discover novel viruses, to identify emergent season and geographical trends, develop protocols to even identify mosquitoes molecularly, transinfection, to test for the outcome of arbovirus and ISV infection, and going to the bacteria, we were able also to produce monocolonization and production of double-strand RNA inside the mosquitoes. So that is a work that is on the way, but I hope it will go somewhere, and it will mostly stimulate people to think at different ways of exploiting all the basic knowledge we have been generated on innate immunity during all these years to fight arbovirus transmission by mosquitoes. So to finish, let me thank all of my lab, especially Cassandra and Otavia that are in charge of these projects, the former lab members that make all the science that now allow us to develop these methods, our past funding that allow us to sequence so much, our current funding, and all of you for your attention. Thank you very much.
Thanks for the great talk. I was wondering, after you say discover the novel strain in the mosquito, is there ability to also predict what virus strain will become more emergent, like viral strain?
Which arbovirus strain will? Okay, so when you sequence sylvatic mosquitoes, yes, you normally don't find necessarily the viruses that have already emerged, because dengue, chikungunya, Zika, they already circulate in an urban cycle. So what you can find are novel arboviruses that have not produced outbreaks yet. So that is possible. However, by sequencing urban mosquitoes and determining the ISV, we also think that we could have markers or predictors. Like if in this season you have this insect-specific virus in the mosquito, perhaps the mosquitoes will be much more prone to transmit or not Zika. So we believe that is possible, but for that you need a lot of modeling and sequencing and calculations.
Thanks, Carla. Great talk. When you sequence the mosquitoes, do you do one at a time? And if you do, can you find interactions between, can you mine that data to find interactions between viruses?
Yeah, we can do pools, or we can do one at a time. We have enough resolution to do one at a time. For viral discovery, we did pools of 5 mosquitoes, okay? But for example, for ribosomal RNA, we did individual mosquitoes for 30 different species. So we have enough data to do the kind of studies that you want. What I don't have is people or money to do it.
That was great. I have a few questions. So I was curious, in the E. coli experiments, so it's really interesting. When you're feeding them the E. coli probiotics, you're looking at double-stranded RNA, but you're not necessarily isolating out the E. coli from the mosquito. So you're just mashing the mosquito, right?
That is a proof of concept. That is why I put the list of the candidates of the local bacterium that we want to modify.
Right. And so in terms of those local bacteria you listed, how many of those are actually tractable? How many of those?
What do you mean by tractable? I sequence them, so they are tractable.
I mean, how many of them are you actually going to be able to
culture?
To culture, but also to transform, right?
We don't know.
Okay.
We don't know. We expect that at least some of them should be modifiable. We're working with bacteriologists in Pasteur, to help us, and we're perhaps thinking that there could be a phage alternative also to all of this, but these are things that we have to discover yet.
Okay. And then, so in terms of establishing a colony, then, if you're infecting them with the one modified genetic host in lab, do you think that'll actually be maintainable in a population outside of the lab?
For the bacteria, you mean, for the double strand? So what we are aiming is that the double-strand RNA will get integrated into the mosquito host, and then it will be already in the genome.
Okay.
So that is our long-term goal. Then you need to think, then you can always modify and put a gene drive system together with your modification, so that when you will do a release, you will have a slow replacement of the natural population, but whatever you are putting in. It's always complicated. Probably you saw in the newspaper that Oxitec is about to release billions of mosquitoes here in California, carrying a protein in the male that when it will mate with the female is supposed to make the female not mature, so the female cannot produce eggs. However, there are a lot of concerns that can really happen and cannot penetrate and modify the natural population.
Right.
So there is always, but there is always a side of the modification that put you in the transgenic scene that has to be carefully measured.
So is that why, what, last one? All right, sorry. It was a really interesting talk. So I was also curious about the genetic incorporation of it then. So it seems like it's probably transposon mediated. So is there a list of insect transposons? I mean, could you just generate a bunch of them for a ton of different viruses, infect your mosquitoes, and have a broad spectrum?
Yeah, so that I
mosquitoes?
didn't show because I don't have the time, but we have already made like cassettes where we just put the LTRs of transposons and pieces of virus in the middle, and you can get expression, yes.
Perfect.
Yes, very interesting all around. But I also sort of related question. When you think about, really getting global control, the presence of this modified mosquitoes in the wild is going to be essential.
Yeah.
And you say that. So there is two things. One is, the fitness of the mosquito when you modify them or infect them. That's the one thing. And I think this is the kind of problems that one see with some of these approaches that you mentioned, including Wolvakia, but also the ones that they sterilize the female. But specifically on your approaches, one is that caught my attention is that you see this seasonality of what virus is in the mosquito population, geographically, but also seasonal.
Yeah.
So it's hard right now to imagine, what will be the perfect virus that you will, infect the mosquitoes and it will maintain over time. So the first question is how do you imagine that the virus will be maintained in the population, is infecting the larvae, or where are you looking for that the bullet, the silver bullet? And the other thing that caught my attention is that, yes, you argue that the double-stranded DNA, the bDNA, will be integrated into the genome, and it will be maintained, in this great approach. But when you look at the passages and the Drosophila, you see that it maintains the effect on the first, say, 10 generation, but at generation 30, you lose the effect. So the question is, what's going on with that? Is it silence? Is it an episome that is lost? What is going on? So the two.
Okay, I start for the second question. That is why you are losing the protection. The trend is still there, but the protection is lost. So statistically, the difference is not significant anymore. So I think of two things. One, the episome could be lost. Two, if it's inserted, but in regions of the genome where it cannot be expressed. Three, we didn't need enough flies to have a statistic value at the population level. When you do control, it will be impossible to target every single insect. You have to think at the population level. You need a certain percentage of insects that inherit and stop transmitting to have an effect, but it's not never 100% penetrance. So that's for this question. But I don't know. I think that probably if it's episomic, I bet that it's lost. And for the silver bullet, yeah, that it will be a terrible study because we really will have to go virus by virus. I think the best solution here will be the transinfection. So as soon as we find a virus where we can show that we have vertical transmission easily in the lab, we do transinfection. So if the mosquito in Cambodia, let's say, instead of using the insect-specific viruses of Cambodia in Cambodian mosquitoes, we will use the insect-specific viruses of Guyana in Cambodian mosquitoes, and then we can control it. But we will have to assess if a change of the virome is not altering fitness, right? Yeah.
Yeah, this was really fascinating talk. Thank you. Not knowing much about the field, I was wondering if you can speculate some. I'm thinking about environmental factors that you can influence by, bringing a new population of mosquitoes into a certain area that take over maybe some diversity. Can you speculate what could be like advantage and disadvantage?
Yes. So the first thing to really stress is that what we are trying to do is a proof of concept in the laboratory. Okay, we're not releasing nothing in the nature. If we have the proof of concept, now is another kind of work that we'll say is totally out of the lab. It's really work that you do with regulation bodies and with ecologists and mathematicians and modelers and that can measure all the kind of effects. Because, for example, you have to measure the effects you will have in water, because remember that mosquitoes have a part of the life cycle in water, and that shrimps will be eating the larvae. So you have to measure so many aspects of the impact that the release of something new can have that is totally out of our expertise. What we are trying to do is to show that there are alternatives to vector control strategy. If one of these alternatives is successful in the lab, we will pair with all kind of governmental and NGO to work into this. I cannot answer otherwise.
Yeah, it's cool. Thank you.
Is there any questions from people on
I
the,
don't have a chat
on
here.
the Zoom? Can they hear? Oh, Jack's got another one? No? Okay. We can talk later with Carla. And maybe I'll ask one quick one. That one species where you said there were so many viruses in there, but you don't know what they are because they're not like any other viruses. There's so many new sequences coming online. You know, are you constantly looking on what's out there, and can you?
Not every day, but our bioinformatician every couple of months match against all these metagenomic data sets. Yeah. And also you have to be very careful because it's more easy to identify an RNA virus than the DNA virus, because the DNA virus could also be an endogenous viral element.
Right.
So you have to be very careful to call something a virus, right?
Oh, there are questions here. Okay.
Okay.
Joe Bodney, can I ask a question from your introductory comments regarding Wolvakia? Given the interest in probiotics as a theme, and despite totally different mechanisms of Wolvakia from other bacteria, could you envision a similar route for failure for both?
Well, I don't like to think of failure in these terms. What I think is that nature is very powerful, and at the end of the day, you create resistance to any modification that you will do. So viruses will counteract, or the insect in itself, in the same way you have insecticide-resistant mosquito, we will finish with Wolvakia-resistant mosquitoes or with my probiotics-resistant mosquito. There is always a way that nature escapes for every control you try to put. So it's not a failure, it's the race for life. I don't know, we just need to be faster and smarter, I guess. So no, I cannot think of that.
Okay, one more here. Swetha. There are so many regulatory and ethical implications to releasing modified mosquitoes in the environment, particularly in developing countries like India. What is your opinion on using double-strand RNA sprays to induce the same kind of effects?
So double-strand RNA sprays were used on insects or in olive trees. Yeah, that was the thing. In Greece, they have a plague, like a little insect that eat all the olive trees. So what they were doing is baby olive trees, and they will put it in under these big containers that will spray with some kind of sand that will penetrate into the leaf of the olive tree. And then this double-strand RNA will turn the olive tree refractory to the virus transmitted by the insect. So it happened. Then with the mosquito, you need to think that it's a complex life cycle that has an aquatic component and a terrestrial component. So you cannot go and spray things like that. I don't think that spraying is a solution for the mosquitoes.
Okay, with that, that's a lot of questions, a lot of interest. Great talk, Carla. So let's thank Carla for a fantastic seminar.
Thank you very much.